the next 2 weeks after this week. Okay? So we've got one more full week of classes next week in this class. So regular, lecture material and things. So next Monday we will do, Dr. Morgan will do some lab intro and some explanation of what you're going to be doing in lab. And then you will be taking your quiz on Chapter 9, Section 3. I've already given you that assignment, but that's pages 183 through 189. Okay? That was originally all the quizzes I was going to give. Okay? And what I was going to do then was let you drop your lowest quiz. What I'm going to do is I'm going to add another quiz, which basically will be optional. Okay? And that will be the Monday before the exam. And that will be on virus, which is Chapter 17. And I'll give you the page numbers for that later on. If you take that quiz, then you can drop your 2 lowest quizzes. Okay? So there's really no reason not to try to take this quiz, because if you bomb it, then you just drop that one as well as one other one. But this represents an opportunity, because let's say you've already got 2 quizzes you've got bad scores on. Well, you automatically can drop one of them, but then that second score could basically be replaced by this quiz score. Okay? Does that make sense? Okay. The exam is a week from next Tuesday. It's not next Tuesday. Sorry, I take some of you to start. So next week we will finish talking about Chapter 16, cancer. We'll then also talk about virus. A week from next Monday, we will have probably some more lab introduction. And then this optional quiz on virus. And then a week from next Tuesday is your exam. The final 2 days of the semester, that last week before finals, Thursday and Friday, will be review. Okay? So we'll be doing some review for the final exam. Okay, here's the breakdown for your exam a week from next Tuesday, your 4th exam. There will be 33 multiple choice questions, 2 points apiece. And here's where they're going to come from. There will be 10 of them from Chapter 14. Now remember that we covered part of Chapter 14 on the last exam through transcription. So it'll be translation and then RNA processing, mutation. And I believe that's study guide questions 9 and above. The first 8 study guide questions were the last exam. Okay? There will be 12 multiple choice questions from Chapter 15, regulation of gene expression. Okay, so chromatin modeling, transcription, okay. And then there will be 11 multiple choice questions from this one section of Chapter 9 and one section of Chapter 16. This is the cancer stuff. Okay? So that's your multiple choice. There will be 4 disagree and agree questions. Okay? And there will be at least 1 from each of these chapters. There will be 1 8-point essay, and there will be 1 10-point essay. Now, additionally, I will have 6 bonus multiple choice questions worth 1 point apiece. And those will be on the chapter on virus. Okay? So if you find yourself really getting jammed up and you want to concentrate just on these chapters, that's okay, but then you probably won't be able to answer those bonus questions. And this is material we will cover in class. I originally was going to include virus within the bulk of the exam, but it's quite a bit of material. So the bulk of the exam will be these chapters, and then if you choose to prepare for the virus section, which we'll cover in class, that will be bonus. Okay? Also remember that you can bring 2 pages of notes, and I consider 1 piece of paper to be a page, so you can use front and back of 2 pieces of paper. They need to be in your handwriting, okay? Because there's a real reason to do this. It's a way to organize your study. But it's not going to help you if you just photocopy someone else's notes. Okay? And then you can also bring the notes from the video that you saw in lab, because the essay questions might relate to that, because that's material we talked about in class: mutation, sickle cell anemia, the ruminative evolution because of duplication of genes, and so that's kind of all relevant. So bring those notes with you. So does that make sense? Okay, can I erase that? I'll start by working over here and give you a chance to write this down and get rid of it now. I hope that the video that you saw on Tuesday was interesting in a number of ways. It is kind of cutting edge information, but what really strikes me every time I see it is that this was a relatively straightforward question that initiated this particular whole area of biology, RNAi. And the initial question was a pretty simple one. How can I make a more purple petunia? And there was basically a profit motive associated with solving that question. Those biotech people were trying to make something that would attract and impress an investor. So they stuck some genes into a petunia plant, thinking that the more recipes for purple they gave it, the more purple pigment it would produce. Well, that seems logical. But they got a very unexpected result. They got no pigment. Okay? So that particular observation resulted in almost a 10-year investigation. And the culmination of that 10-year investigation was this new hypothesis was that putting genes into a genome could silence some of the other genes, could turn them off. And so that genetic material could regulate the expression of other genetic material. This has really, really important applications, both of which they talked about in the video. One being, if you can specifically make little segments of genes that then can turn off other genes, then you can correlate genotype and phenotype. I'm going to knock out this gene and then see what happens to the organism. Then I know something about what that gene does. So we call those knockout experiments, and those are very fruitful. But then also the medical applications are really intriguing in how we might use this RNAi to treat various genetic diseases. The woman with macular degeneration, okay, she was making too many blood vessels. And so can we somehow knock out the genes that code for too many blood vessels being produced? And they seem to be getting some good preliminary results from that. So what seem to be kind of innocuous questions that people are trying to answer sometimes turn out to take really unexpected turns and have really kind of groundbreaking applications associated with them. So stay tuned. You will be hearing more about RNAi. It's really an important development in biology. One of the things that's really interesting in an application of what we've been talking about is that researchers are now able to monitor expression of specific genes in either living organisms or tissues of those living organisms. And that's opened up whole new areas of study which have become very fruitful. Remember that in multicellular organisms, cell types are different from one another, not because they have different DNA, they've all got the same DNA, but because different genes within that DNA are being expressed in different tissues. So there's differential gene expression. So what's the most straightforward way to discover which genes are being expressed in different cells? Well, looking for the messages that are being produced. Okay, because if gene expression is going to be occurring, messages are going to be produced, and if those messages are then translated, proteins will be produced. So there's an important tool that's used in these types of studies, and also this tool is the basis for other DNA technologies. And this is called hybridization. Let's take a very simple example. Let's say we've got two genes. We're just going to call them gene 1 and gene 2. Okay? And these two genes, this one has a stretch of adenine nucleotides in it, and this one has a stretch of guanine nucleotides in it. Okay? Well, how can we determine whether those genes are being expressed? Well, we have the technology available to make what we call probes. And probes allow us to kind of identify genes. So let's say we make a probe, and this probe is a piece of single-stranded RNA, or it could be DNA as well. So we make this little probe, which is U. Okay? And then furthermore, we put a label on this probe so we can detect it. In this case, it might be a fluorescent dye that fluoresces red. Okay? So how can we identify which gene is being expressed if we have this probe? Well, we mix the probe with the genes and allow them to hybridize. What hybridization means is that they bond together. And how do they bond? They bond by complementary base pairing. So if we mix this probe with these two particular genes, and this probe represents the messages for gene expression, then which one is the probe going to bind to? It's going to bind to this one, right? So we will get this hybridization that will occur. And then you can rinse the tissue to wash away unbound probe. And then what we would see is that this gene would glow red, and this one would not, because it didn't hybridize with the probe. So this particular probing, using this hybridization technique, has become a really powerful technique that's allowed individuals to study gene expression in tissues and living organisms. Hybridization is also the basis for other technologies, including polymerase chain reaction, which is a technology that you read about in your quiz last week again. So this idea of DNA hybridization is a really powerful and important tool. A couple of examples. Okay? Here's an example where we're using hybridization in a living organism. This is an embryo of Drosophila, which is the fruit fly. And this particular embryo has been incubated with 5 different probes for 5 different messages that researchers are interested in, and they want to see which cells express those particular messages. So they've labeled some of the probes yellow, some blue, some green, some red, etc., purple. Okay? And then they incubate the organism with these particular probes. And they see where do the probes attach, which cells are expressing this message, which cells are expressing this message. And this shows up in the various colors. So these individuals are very interested in studying the embryological development of the fruit fly and seeing which parts of the fruit fly develop into which structures. And that's really the result of which cells are expressing which genes. This particular type of hybridization is called in situ hybridization. That means in place. And literally, we're using living tissue in place and soaking it with these probes and then seeing where the probes hybridize and therefore which genes are expressing which messages and which proteins are being produced. Almost looks like artwork. I would have thought that Nina Tandon, who came here a couple of weeks ago to speak, would have liked this and talked a little bit about how molecular biology can be art as well as fabric and materials. There's another hybridization technique that's really cool also. Called DNA microarrays. Also referred to as DNA chips. And in these DNA microarrays, genes are fixed to a piece of glass. And that glass has little indentation or wells in it. In the one I'm going to show you in just a second, each well has a gene from 2 different tissues. For example, One of the tissues might be breast cancer tissue, and the other might be normal breast tissue. They then can construct probes for those genes. And they're labeled with fluorescent dyes. So let's say that the breast cancer probes are labeled red, and that the normal breast tissue probes are labeled green. So they incubate these DNA chips with these probes, and then they can determine gene expression in those two different tissues. What's being expressed in the breast cancer tissue? What's being expressed in the normal tissue? And that's going to give us some real insight into what changes have occurred in cancerous cells versus what changes have occurred in normal tissue. So here's one of these gene chips. Okay, this is the DNA microarray. These are the little wells where the genes are fixed. This particular microarray has about 5,700 little wells. And each well has a gene, a specific gene from a normal tissue and a gene from a breast cancer tissue. Okay? That's about a quarter of the human genome. Okay? There's about 20,000 genes. This is about 5,000. The way you interpret this is that areas where it is black, neither probe has attached, and therefore those genes aren't being expressed. Genes in yellow are being expressed in both tissues. Genes in red are being expressed in the first tissue. For us, that would be the breast cancer tissue. Genes in green would be expressed in the second tissue. That would be the normal tissue. So this would allow you to start honing in on particular genes and saying, this one's being expressed in the normal tissue, but it's not being expressed in the breast cancer tissue. This one's being expressed in the breast cancer tissue, but not in the normal tissue. And we can start identifying, therefore, genes of interest that we want to study more in determining whether they're implicated in cancer. So these are very powerful tools. I think kind of one of the holy grails of medicine is that all of us will have in our medical records at some point in time a gene chip for all of our genes. And the doctor will have it on file, or you'll bring it into the doctor. And they can actually determine then how you respond genetically to various drugs. Not all drugs work the same on all individuals. There are certain populations of people, for example, that react very poorly to certain types of blood pressure medication. Well, do you have that gene or not? Pop your gene chip into a machine and determine whether you have that gene. And then the doctor can tailor your treatment to your particular genetics. They call that personalized medicine. Sounds very cool. See any downsides? Is your doctor having all of your genes?
If your genes are in the wrong hands, like a massive population, that can be a genocide.
Yeah, I mean, do you want people knowing what all of your genes are? What if your genetic information, your gene chip, shows you that you have an inherited predisposition to certain types of cancer? Yeah, what if your insurance company's got a hold of that? Okay. Could they then deny you coverage because you have a genetic predisposition to a disease that could cost them a lot of money? So there's lots of issues here about confidentiality, sharing information, who has access to it, who owns it. So like any new technology, these particular advancements have both positives and negatives. If you're interested in these kind of issues, and I think everybody should be, Dr. Rhodes teaches a bioethics class, and he talks a lot about bioethical issues of technologies in biology, reproductive biology, stem cell, etc. And it's a really interesting course that really stretches you to think and to questioning some of your assumptions and beliefs. Okay, so let's move on to the last kind of quote-unquote required information for your next exam. And that's cancer. Okay? In the textbook, they have the material relating to cancer split up into 2 chapters. Chapter 9 is the chapter that deals with mitosis and the cell cycle. And I understand you covered that chapter last semester in 134. You probably don't remember it. You might remember the mitosis part, but the cell cycle part, probably not so much. So that's what the quiz will be on Monday, and that's kind of foundational material for understanding cancer. So that's 9.3, pages 183 through 189. Chapter 16 in your book talks about some consequences of genes being expressed in different manners. And the first part of the chapter talks about embryonic development. And we just don't have time to get into that. So we're not covering that part of the chapter. We're going to really cover the last section of chapter 16, which deals with what happens when genes are expressed inappropriately. What happens when there are mistakes in gene expression? And the result of that is cancer. So we'll talk about cancer. So that material will be spread through these 2 chapters. Okay, so that's why over here I put chapter 9, 16. Okay? So here's a cancer cell. This particular cancer cell has some unique characteristics associated with it. One of the things that oftentimes you'll see with cancer cells are all of these little kind of projections that stick out from the surface. They're called blebs. Not all cancer cells look that way, but this is an example of some of the morphological changes that can occur within cancer cells. There are all kinds of changes that occur within cancer cells. Number of chromosomes can be different. The metabolism of those cells can be different. But one thing that seems to really be common to all cancer cells is that they have lost the ability to regulate normal cell division. And cancer cells start dividing out of control. And once they start dividing out of control, there are real consequences for the organism. How many of you know someone who has cancer? Pretty much everybody, right? In North America and in Europe, about 1 out of 5 people will die from cancer. You look around the room, and that's a little scary. I mean, several of us are going to die from a cancer. And we also see that the risk of cancer generally increases with age. Any idea why? Why is your risk of cancer higher when you're my age than when you're your age, Jenna? Absolutely. There's been more time for your cells to accumulate mistakes or mutation. Jesse, what were you going to say?
I was going to ask if anything to do with telomeres.
It could, yes. We didn't really talk about telomeres that much, but shortening of telomeres can have an effect. Another thing that happens is as we get older, our immune system declines. What's the immune system got to do with it? Ideally, if a cell becomes cancerous, oftentimes as enough changes occur within that cell that the immune system recognizes this as being a threat. And it responds by killing that cancer cell. All of us within our lifetime have genetic changes occurring within our cells that result in cells becoming cancerous. But for most of us, most of the time, those cells that become cancerous are quickly eliminated by the immune system before they start to proliferate and cause problems. One of the interesting questions about cancer is, why is it that some types of cancer seem to evade detection by the immune system, and are allowed to persist when the immune system should eliminate them. So there's a lot of overlap between the study of cancer biology and immunology. Cancer cells can be thought of to exhibit antisocial behavior. What does that mean? Well, cancer cells are bullies. And they literally bully other cells, and they push them aside, and they dominate them. Cancerous tissue can grow into normal tissue and destroy it. Cancer cells invade others' neighborhoods. Cancer cells don't communicate with other cells well. One of the things we're going to see is that in normal tissue, there's a phenomenon that occurs called contact inhibition. And once a cell contacts another cell, some signals are sent that says, you're here, I'm here, let's stop growing. And that basically cuts off cell division and keeps one cell type from overgrowing. Cancer cells don't communicate with the other cells well, and they don't show this phenomena of contact inhibition. They just keep growing and growing and pushing and crowding. That's a problem. Cancer cells don't respond appropriately to environmental signals. And we're going to see in a minute that there are a couple kind of signals that normally regulate cell division. And some of them are internal signals. And cancer cells don't respond well to those. But some of them are external signals too. There are substances called growth factors, which normally are needed to stimulate cells to start dividing. And many times cancer cells will divide even if the growth factors aren't present. They're responding inappropriately to those signals. And they don't obey the rules of society, like dying when they should. Normal cells have a limited number of cell divisions that they can go through, and then they start dying. That's one of the reasons why we age. But cancer cells are more or less immortal. They just keep growing and growing and growing and growing past the time when normal cells should have died. So, what's causing cancer? Well, cancer is fundamentally a genetic disease. So we've spent a fair amount of time studying genes. So now we've got a little bit of background to kind of understand what might be going on genetically within a cancer cell. What causes these genetic diseases? Well, they can often be caused by spontaneous mutations. Remember that those are errors in DNA replication. And in this case, the cells fail to repair those errors. So the repair systems in cancerous cells might be malfunctioning. They can be caused by induced mutations, various environmental factors. Environmental factors could include various chemicals, or they can include various types of radiation, like ultraviolet radiation. Remember, ultraviolet radiation can cause skin cancer. Maybe some of the chemicals that you consume in your foods are potentially carcinogenic. We talked last time, one of the last classes, about how there are some screening tests that can be used to screen food additives to see if they might cause cancer by determining whether they cause mutation in bacteria. Remember the name of that test? It's called the Ames test. Okay, that's that screening test to see if things are cancerous by screening to see if they cause mutation. What's really interesting is that we now know that there are certain types of cancer that are caused by virus. We now have a vaccination for the HPV virus. Anybody know what HPV stands for? Human papilloma virus, okay?
And if you are infected with this human papilloma virus, women, for example, have a much higher incidence of cervical cancer. And so we now have an HPV vaccine to try to prevent that type of cancer. So the idea of an anti-cancer vaccine is an interesting concept.
Well, the genetic mutations that cause cancer are usually part of some key regulatory pathways. And these are pathways that are involved in generally kind of three categories. A lot of them are involved in pathways that control cell division. Some of them are involved in pathways that control cell growth. And others are involved in pathways that are involved in response to DNA damage and stress. So some of these might be DNA repair pathways, for example. So if genetic mutations occur in the genes that regulate any of these pathways, you've got a potential for a cell to become cancerous. A particular nasty type of virus, glioblastoma, which is the most common brain cancer, has actually been found to involve mutations in all three of these pathways. So all three of those particular phenomena are being damaged at the same time in that particular type of cancer. So, what happens in cancer development? A normal cell becomes transformed by either mutation or viral infection. So its genes have somehow been altered. Okay? You transformed E. coli in the lab by adding pGlo genes to it. Okay? And therefore you change the genotype of that cell. Your cells could be transformed by having viral genetic information added to them, or various chemical agents or radiation might have caused mutation within genes, or errors in DNA replication might have caused mutation. But a cancerous cell has become transformed. Its genotype has been changed. When the cells become transformed, they start dividing rapidly and out of control, and they form masses of abnormal cells called tumors. A tumor might remain at the original site. And if that's the case, it is typically called a benign tumor. Okay, the cell's still been transformed, but the cancer tissue isn't invading surrounding tissues, for example. What's typically the method of choice of treating a benign tumor? What do we do to it? We remove it. Okay, so surgery, surgical removal. And oftentimes that is also accompanied by some radiation treatment, because when you cut out a tumor, you're not always sure that you've gotten all of the tumor. And cancer cells that seem to be particularly sensitive to radiation, in part because their DNA repair mechanisms might have gotten screwed up, and so they can't correct defects caused by being exposed to radiation. So benign tumors are usually pretty treatable, and the prognosis of recovering from those is very high. Now, if a cell accumulates enough genetic changes, and the right ones, then that transformed cell can become malignant. And it can invade the surrounding tissues, and then also some of the malignant tumors can metastasize. They start releasing their blood cell, their cancer cells into maybe the circulatory system, and a brain tumor can metastasize to the lungs or the liver. And so multiple organ systems in your body can be affected by that same cancer as it spreads throughout the body. Typically, we don't refer to these benign tumors as being cancers. Typically, we refer to things that are malignant and metastatic as being cancers.
How do we treat these? Is surgery and radiation usually effective for these malignant metastatic cancers? What's the alternative treatment we usually use in these situations? People that you've known that have had cancer, if they haven't had surgery or radiation, what are they probably having? Chemotherapy. And there are various anti-cancer drugs that can be given intravenously, for example, and can be used to try to selectively kill the cancer cells. Are those chemotherapeutic drugs usually completely selective? Do they sometimes kill normal cells? Yeah, they do. And that's why we can have some of the side effects of chemotherapy. Okay, normal intestinal cells can be killed by the chemotherapeutic drugs, giving you nausea and vomiting. Hair follicle cells can be affected. Your hair falls out. Okay, so chemotherapy is really kind of a nasty treatment, and we're constantly looking for more targeted ways to deliver these killing drugs just to the cancer cells and try to preserve the normal cells. And that's a real challenge. So here's a tumor that's developing because a cell became transformed, its genetics changed, and it starts dividing out of control. Okay? If that remains localized and is benign, then it can usually be removed surgically, maybe some radiation treatment. But if it starts being invasive, which usually requires additional genetic changes, then it can invade surrounding tissues, and then if it metastasizes, those cancer cells can be transported to different parts of the body. And that is the worst case scenario. So, how do we begin to get a sense of understanding all of this? Well, the easiest place to begin is to look at this idea of cancer cells grow out of control. They don't seem to be subjected to the normal regulations that tell cells when to divide and when not to divide. The cell cycle is that sequence of events that a cell goes through in its lifetime from one cell division to a next. And the cell cycle is regulated by a molecular control system. Now, do all cells divide at the same rate? No. We've got some cells in our body, like our epithelial tissue, which pretty much constantly divides. Why does your skin epithelium constantly divide? Yeah? Pardon me? Yeah, I mean, your skin cells are constantly dying. They're being sloughed by the surface of your tissue. Most household dust is dead skin that you've sloughed off. You've got to constantly replace those cells because you're constantly losing them. Same thing is true with your intestinal epithelium. You're constantly losing those cells and they have to be replaced. What are some cells in your body that typically don't divide? Nerve cells typically don't divide, and that's why nerve damage can be permanent. That's really a very fruitful area of study, is spinal cord injuries, and are there ways we can stimulate nerve cells, which are kind of dormant and not dividing, to start dividing and start repairing the damaged tissue again. So why do different cells divide at different rates? Well, there's an internal regulating mechanism within the cells that operates at the molecular level. So if we can kind of understand how this normal regulatory mechanism works, how cells get the proper stop and go signals at the proper time, then we can get a better insight into cancer because that regulatory mechanism has been disrupted. And they're not getting the proper signals any longer. They're just going go, 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 and never being told to stop. in some respects, cancer is the price that organisms pay for having tissues that can renew and repair themselves. Okay? If cells didn't divide, then cancer wouldn't happen. So let's look a little bit at the cell cycle control system. And the analogy that they make in this is that it's kind of similar to a clock. The clock, the cell cycle, has specific checkpoints, and these are key places within the cycle where the cell gets stop and go signals, and those signals determine whether it moves on through the cycle or whether it stops during the cycle. These are called checkpoints. And these checkpoints can be influenced by both external and internal signals. And we'll talk about what a couple of those signals are. So, here's our basic cell cycle control system. Okay? So this represents kind of the life of the cell from one cell division to the next. Remember that the cell cycle is broken up into 4 phases. There's this G1 phase, the S phase. Remember what happens in the S phase of the cell cycle? What has to happen prior to mitosis and meiosis? Replication of the DNA. Okay? So during this S phase, all of that DNA replication stuff that we learned about, leading strand, lagging strand, Okazaki fragments, polymerase, that's all happening in this part of the life of the cell. Okay? Then the G2 phase is kind of a preparatory phase for those duplicated chromosomes to be pulled apart in mitosis. And ultimately, the nucleus divides, the cytoplasm divides, and by the time you finish the cell cycle, one cell has divided to form 2. Okay? How long does the cell cycle take? Well, if you're a bacterium, about 20 minutes on average to complete the whole cell cycle. In eukaryotic cells, vast difference in times of the cell cycle depending upon the cell type. Okay? Probably the fastest ones, take 18 to 24 hours. But some cells never really complete the cell cycle once they're formed. Okay? They don't divide again, like nerve cells. So these checkpoints are kind of important parts of the cycle that determine whether you proceed to the next step or not. It's almost kind of like a gate. And if the gate is open, then you can pass the checkpoint and move on to the next one. If the gate is closed, then you can't move past that checkpoint. For most cells, this G1 checkpoint seems to be the most important one. Okay? This is kind of the first hurdle of cell division. And if the cell receives a go-ahead signal at that checkpoint, then it continues on. And it continues on into the other phases until, if it passes this checkpoint and this checkpoint, it finally divides. If a cell doesn't receive the proper go-ahead signal at that first checkpoint, then it kind of exits the cell cycle. And it switches into a non-dividing state called the GO state. Okay, so here's the G1 checkpoint. And if this checkpoint is open, then cells continue through the cell cycle and will ultimately divide. But if it doesn't get the proper signal, then that checkpoint is blocked, and the cell basically just kind of goes into this arrested state of cell division. So this would be a nerve cell, for example. Okay? This also might be a liver cell. Liver cells typically don't divide once the intact liver develops, unless there is damage to the liver tissue. And that damage can send a signal to liver cells, let's move out of this GO phase, and those signals then allow it to pass the checkpoint, and liver cells can then start dividing again. That's why you can do a partial liver donation to someone that you match to, is they can surgically excise part of your liver and then that liver tissue in your body will grow and replace itself, and the transplanted piece of liver you give can regrow and establish a new liver in the donor as well. Well, there are some both internal and external signals that determine whether those checkpoints can be passed. An example of an internal signal is whether the chromosomes attach properly to the spindle fibers. And if they don't attach properly, then the cells won't move past that M checkpoint, and the chromosomes won't separate. What I want to talk about next time are some of the external signals that influence whether you can pass checkpoints. And some very important external signals are these substances called growth factors. And typically these growth factors are needed for cells to divide properly, particularly if they're growing in tissue culture systems. These are systems where you can grow cells outside the body in complex media, kind of like you grow bacteria on agar plates or in broth cultures. So that's kind of where we'll pick up next time to talk about some of these growth factors. Okay? So Monday, lab information, anything they should be reading? Next lab section? Next lab section. And then quiz over this material, okay?